In this lab, we will revisit how the effectiveness of antimicrobial drugs can be measured in the lab. Let me remind you about the term minimum inhibitory concentration, otherwise known as MIC. This is the lowest concentration of a drug that inhibits the growth of a microbe. Two versions of a dilution broth test that is used to measure MIC are shown on this slide. On the left here, these are just test tubes in the lab where the same amount of bacteria was put into each test tube, but then an increasing concentration of drug was added to each tube. So this tube has 2 micrograms per milliliter, then 4, then 8, 16, and 32 in the tube in the far right. And you can see just with your eyes that this lower concentration tube is very cloudy. And as you go up to 4, it's maybe a little bit less cloudy. And at 8, all of a sudden it's not cloudy. 16 is not cloudy and 32 is not cloudy. And so in this case, 8 micrograms per milliliter would be the minimum inhibitory concentration for this type of bacteria with this type of drug. This kind of assay can be miniaturized into a microtiter plate, like this one is shown here, where you can put a number of different drugs and organisms all into the same plate and measure the cloudiness of the media as an output of growth and determine the MIC. So for example, if we look at this row here, this is a row that has erythromycin in it. This one has growth, 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 growth. Here it's finally clear. And this concentration of drug here was 8 micrograms per milliliter. And so that would be the MIC for erythromycin for this particular microbe. For penicillin, it's 0.06 micrograms per milliliter. And for clindamycin, here at the very top, clindamycin was never able to prevent the growth, even at the highest concentration of drug used, which was 32 micrograms per milliliter. Now, we don't say that that means that clindamycin is completely ineffective. We just say that the MIC is greater than 32 micrograms per mil because that's the highest concentration we tried. But maybe if we'd gone up in concentration, eventually an MIC would be found. Another way to look at Another way to look at the effectiveness of drugs is to look at the zone of inhibition around a disc containing the drug that is put on an agar plate with a lawn of bacteria. So you can see in this diagram here, here's the plate. This yellow color is representing the bacteria that are growing. These darker yellow circles are the antibiotic discs. So each of these discs has a different antibiotic on it. And this white zone here is a zone of clearing called the zone of inhibition. So the way the zone of inhibition is measured is by using a ruler to measure the diameter from one point to the other across the zone of inhibition, as you can see here on these actual plates. Minimum inhibitory concentration can also be measured in a similar assay to the Kirby-Bauer disc diffusion assay. This is called an E-test, where instead of a disc with one concentration of drug, you have a strip with a gradient of drug. So up here on this part of the strip, it's around 256 micrograms per milliliter. And as you move down the strip, the concentration is decreasing. So up here, the concentration is higher. You see this growth, the zone of inhibition. But as you decrease in concentration, that zone gets smaller and smaller and smaller until finally you find the minimum inhibitory concentration, which in this case is about 1.5 micrograms per milliliter. For today's lab, you read a story about a woman named Marissa who has a urinary tract infection. The drug that she was given was not effective, and so now the bacteria are being tested for susceptibility to different drugs. Here's a plate with the bacteria growing on it that were cultured from the urine specimen with a number of different drugs, which are labeled A, B, C, D, E, F, G, etc. And the zone of inhibition around each has been measured for you. So what you will do is take these measurements for each drug and fill in the table provided with the diameter of the zone of inhibition. And then, using these measurements here, you're going to determine whether the bacteria is resistant, intermediate, or sensitive to each antimicrobial drug. If the ZOI is less than the number in the resistance column, then you would mark the bacteria as resistant to that particular antibiotic. If the ZOI falls in the intermediate zone, you would call it intermediate. And if it falls in the sensitive zone, then you would determine that the bacteria are sensitive to that drug or susceptible to that drug. And you might notice that different drugs have different zones of inhibition boundaries. So, why is that? Why is a zone of inhibition of 16 considered resistant for gentamicin, but a zone of inhibition of 21 would be resistant for erythromycin? Well, it's because these drugs diffuse differently through the agar based on their size and chemistry. Some diffuse further than others. And so you can't just use one standard boundary for all of the different drugs. So once you've filled out these last two columns in the table, then you'll answer the questions that are on the assignment page and submit them on Canvas. Hopefully in your study, you'll find at least one antibiotic that seems promising to try for treating Marisa's urinary tract infection.